Okay, so let me just, if you don't mind, I'm just going to reintroduce myself so we have it for the recording. My name is Anne-Marie Della Pizzi. I am on the faculty of Dominican College. I'm also the Honors Program Director, and I am the faculty representative to the NRHC Executive Board. So welcome to the conference. Welcome to this session, which is Ecology 1, Ecosystem and Human Health. So we have three presenters for this session. We have Ana Acosta from Dominican College, Gabrielle Biseinere from Westfield State University, and we have Hinal Rana from the University of Hartford, and we'll be presenting in that order. So I think what we'll do is, because we have a little bit of extra time to play with in this session, each student will have 15 minutes to present. And then we will have about 5 minutes of questions after each presentation. And then that will still give us a little bit of time at the end if we have, final questions or maybe questions for the group. Also, please feel free if you have any questions that you would like to type into the chat room. Please feel free to do that, and I will read some of those questions at the end of each presentation and perhaps even when all the presentations are done. Okay, is everyone good with that? All right, so then without further ado, I'm going to turn the podium over to Ana Acosta. She's from Dominican College of Blauvelt, and the title of her presentation is going to be Glyphosate Pollution in Residential Communities with Bacterial Analysis.
Thank you for that introduction, Dr. Della Pizzi. Let me just share my screen real quick. All right, so as she said before, my research is on glyphosate pollution in residential communities with bacterial analysis. My name is Ana Acosta, and I come from Dominican College. First of all, I just want to point out on this slide that glyphosate is actually an herbicide, and it's commonly found in Roundup. So just so you have that background to start off with. Over here, you can see some pictures that I have of the Piermont Marsh, which is where I was focusing my research about, because the New York State Department of Environmental Conservation, they proposed a plan to treat the Piermont Marsh with glyphosate, and they proposed that in order to treat the invasive monoculture of the Phragmites, which you could see in the middle picture there. So they wanted to do that since the Phragmites is a very invasive monoculture and it decreases the biodiversity in that area. And you could see that the marsh runs along the west side of the Hudson River, all the way up to like the bridge, which there's a lot of heavy traffic. There's about 2,540 residents in Piermont. A lot of them were fairly opposed to this plan that was proposed. And there's also a lot of tourists that go to this area too. I don't know if you guys have ever heard of the Piermont Pier, but I mean, I go there all the time to fly kites, so it's a great place. So the purpose and the goals of this experiment was mainly I wanted to see what impact the glyphosate had on the microbial community structure. And I wanted to then see how the antibiotic resistance changes on those bacterial communities with the glyphosate. So the goals were to develop a model of the environment treated by the herbicide, and then to test the antibiotic resistance. It turned out to be Pseudomonas, which I'll explain later on, and to test the antibiotic resistance of the Pseudomonas from treated and untreated samples. Continuing forward, this was my procedure basically. I had the sediment samples collected and I split it into 4 jars. One was untreated, one was 2.75%, which is the low concentration. Another one was 3.9%, which is the medium concentration. And then I also had the 5.25%, which is the high concentration. And every week for 3 weeks, we added glyphosate. And then at the 3rd week, we cultured samples from each concentration. We cultured onto EMB plates, which grow gram-negative bacteria. And then I allowed the samples to sit for 3 weeks and cultured again at week 6. I did not add any more glyphosate during those last 3 weeks because I consider that to be like a recuperation period to see if it can go back to normal or not. And from the cultured samples, I used antibiotic discs to test against a bunch of different antibiotics. I just want to add a little something here that we also collected some of the sediment samples and sent it out for next generation sequencing to see the entire microbial community. And that's like from the samples themselves. Let me see if I can get a laser pointer. So from these samples here at week 3 and at week 6, we sent it out. And I also took some of the isolates from the cultured plates over here and sent those out for sequencing as well. Over here, you could see this is the untreated cultured plates. It is labeled by week. So week 1 is always going to be untreated going forward because that's the baseline. Then you have week 2 and week 3. This is untreated again, so there's not much change in the growth. And then after the recuperation period, not much change either. And I have a bunch of different serial dilutions that we did as well. You could see over here just the difference between the untreated and the low concentration. There was a huge increase in the growth of bacteria. And the same thing for the medium concentration. There was a huge growth. I'll let you just take that in for a second. And finally, this was a high concentration, and you can see over here, this plate specifically was capsulated. So what that is if you take a toothpick and you pick up the bacteria, like it'll just hang from there. And you know, bacteria is small, so having that is pretty interesting to note. Also, another thing was I included pictures of the sediment samples themselves. So I have the untreated over here. You could see it looks basically the same as the marsh water. It's not much different. Then I have low concentration, and I want to point out this like yellow thing. You could see like the layer of it. That's called a biofilm. And that's basically a layer of bacteria that all gets together in that tight-knit community. And it goes like that to thrive, but it, you know, like the bacteria end up sharing nutrients and all that. In the medium concentration, you can kind of make it out as well. You know, it goes around, it's completely different. You can't see through the water, you just see the biofilm. And in the high concentration over here, you can see there's a very thick biofilm. And another little thing I want to add here is that the dirt smelled horrific. And my classes were actually in the same building that I was doing this research in. So after every class, I would tell my friends, I was like, hey, you guys want to go smell my dirt? It was a lot of fun, but it smelled bad. So going further this is the information that I obtained from Purple Line on DNA Subway from the sequencing that was done for the sediment samples, not the cultured ones. So this was the entire, we sent out like a little vial of the dirt at different concentrations, which you see over here, and different weeks. I want to point out specifically the Pseudomonas. This is broken down by the genus level. So you can see there's a huge increase in the Pseudomonas as time goes and as concentration increases, even after the recuperation period, which is the most important thing, is that once you add Roundup, even after recuperation period, it doesn't change back to normal. It continues to enhance bacterial growth. And from that information itself, I actually also got the Pseudomonas count for each of the samples. And I ran an ANOVA test to compare the untreated against all of the week 3 concentrations against all the week 6 concentrations. And there's no statistical significance because when you compare the p-value to the alpha, which is 0.05, the p-value has to be less than the alpha for it to be statistically significant. So there was no significant difference with the ANOVA. However, you can see when I did the t-test against the untreated versus all of week 3, it was statistically significant. So that was different. And then again, with the untreated versus all of the week 6, there was no significant difference with that. Going further, this information came from the cultured isolates that we sent out. So that's from the actual plates themselves. And you see, we did a blast N search, and it just confirmed the information that what we were growing was Pseudomonas. And that was important because that helped us determine which antibiotics to test against. So the way we did the antibiotic testing was using these discs, which produce a zone of inhibition if it has any effect on the bacteria. And so the way you measure that with the zone of inhibition against the antibiotic resistance is you measure the diameter. I measured in millimeters, and then you're given a key by the company who you get the discs from of the susceptibility or the resistance, what you need to be resistant. And I also use these strips for what's called an E-test. And what that gives is the actual dosage to become resistant to the bacteria. Over here, I just pointed out all the antibiotics that I used, and then I have a key over here to see which ones are resistant and which ones are susceptible. It's broken down by the weeks, concentration, and all that. Oh, one thing I wanted to note here, sorry. The penicillin and the novobiocin were completely resistant with all the different bacterias and strains and all that. So there was actually no zone of inhibition. The bacteria just completely grew over that. Over here, I have the p-values for ANOVA tests that were done for the different antibiotics. What I want to point out here is that for week 3, this was comparing all concentrations in week 3, and there was no significant difference. However, once you factor in week 6 and compare all of those with an ANOVA test, then you have a significant difference for streptomycin, kanamycin, and clindamycin. This one was for the E-test. It shows the value for that. It shows the differences in the dosages needed for week 3 and the dosages needed for week 6. You can see there's like a slight increase in all the dosages, except for kanamycin. And some of the conclusions for this were basically that there was a huge increase in the bacterial growth in sediment samples. The antibiotic resistance was increased with a recuperation period for certain antibiotics, but with other antibiotics, there was just no significant difference. Some of the future studies I would love to do with this project would be to investigate which concentrations of glyphosate would actually cause a significant difference on the antibiotic resistance. And then the reason why Pseudomonas is so important to note here is because it actually contributes to greenhouse gases with the glyphosate metabolism. So because you're adding the glyphosate, it breaks it down and then it causes the greenhouse gases to be released. And we all know climate change is a big issue right now because we want to save the earth, and that's why ecology is important. And that's why the Pseudomonas is important, and the glyphosate is enhancing that growth and causing these issues. And it's also a potential human pathogen because it could be related to pneumonia. I want to give an acknowledgement to Dr. Bernadette Connors. She was my mentor throughout this whole project. She dealt with the smell of the dirt with me, and she's just an amazing mentor. She's an amazing professor. I also want to thank Professor Margaret Mulligan. She's a statistical magician. She's a genius with numbers. She helped me analyze all the data because there was a lot of data. And I also want to thank Siobhan O'Sullivan. She's the lab manager over at Dominican College, and she dealt with the dirt smell much more than everyone else did. So I just want to say a special thank you to her for organizing everything and keeping the lab running. Also a thank you to Stephanie Moncayo and Alexis Guillermo. They contributed to the project in different ways. And lastly, thank you to the science department of Dominican College because they helped with funding, and I couldn't do science without them. And I have time for questions. This is just a fun little picture of me getting the sediment samples near the marsh. And thank you for your time. I'll stop sharing my screen and answer the questions. Gotta get the laser pointer off. There we go. So any questions?
Thank you, Anna. So we have some time for questions.
I have a question. Can you hear me?
Yeah, now we can hear you.
So what do you think are the policy implications of this? Because I'm suspecting that you are thinking about, what are the next steps of these findings?
So this research was done in 2019, and that was when they were proposing the bill. What I forgot to mention is that Governor Cuomo actually signed a bill recently, like in 2020, to prevent the use of glyphosate in all state public areas. So it's not really an issue anymore. However, it is an issue on a smaller scale where people will still be using glyphosate on their driveways, and then it goes into the runoff water and it still has the same effect when it all comes together like that.
Thank you.
So what's your take home message to the people in the community? What do you tell them, right? If you have to take this, scientific study and break it down to the everyday average person, how do you explain this to them?
That's a good question. I would just start by showing like the research, like, hey, look at this bacteria. It's really gross. Look at the dirt. It stinks. You don't want that. You want healthy dirt so you can grow plants. I think they would understand that.
Besides Pseudomonas, was there any other significant findings in terms of the types of bacteria that you saw?
We didn't really focus too much on the other stuff because what we did was we had a bunch of serial dilutions, like we went to 10 to the -5. So what we tried to do was streak plates that had similar looking colonies in order to try to keep it, along the same lines for the experiment to keep everything correlated. So we didn't look too much into different bacterias. Anyone else have any questions?
I do. So with the bacterial analysis, did you notice like in the sediment, like different foliage growing in the areas where glyphosate was more present? Like,
Yeah, well, it was the biofilms that grew more. Like, because I already had the dirt collected, the samples, so they were just sitting in the jar the entire time. So it wasn't like anything getting in or anything going out. It was more of just adding the glyphosate and having that be the only factor that's changing throughout time.
Cool. Thank you.
Any other questions or we're good?
Any final questions for Anna? Oops, it's the timer. So any final questions for Anna?
All right, thank you all for your
Okay, great.
time.
Thank you, Anna. Okay, so we're going to move into our next presentation, which is Gabrielle Biseinere from Westfield State University. And the title of her presentation is The Plastic Crisis, Assessing the Impacts of Secondary Microplastics on Cell Proliferation and Apoptosis in the Daphnia Magna Intestinal Epithelium. So without further ado, I turn the podium over to you.
Thank you so much for that introduction. I'm just gonna get set up here. Oops, I clicked the wrong thing. Okay, here we go. Can everyone see and hear me okay? You know what? Forgot to start video as well. Okay, can everyone see and hear me okay? All right. Cool. So just to introduce myself, my name is Gabrielle Biseinere. I am a senior at Westfield State University in Massachusetts, and I am a biology major. My study focused largely on microplastics and the effects that it was having on a small water flea known as Daphnia Magna. This is just an outline of what I'm going to cover today, but I'm going to jump right into it. So in choosing this topic, the plastic crisis is an issue that feels like an impossible problem. There's so much of it and fewer places for it to go. Microplastics are a recently recognized issue. In 2015, the FDA banned the manufacturing of microplastics as spheres that were being put into beauty products. They were finding that it was polluting our fresh water sources, and they didn't have many solutions on how to filter it out properly. And beyond that, it is going into our fresh water, and there are microorganisms and invertebrates consuming them, which is leading to whole populations declining and disruptions of community interactions. And for the purposes of our study, we defined microplastics as being 5 millimeters or smaller in size, and they can be characterized by size, origin, polymer type, shape, and color. And beyond that, there's several categories: fragments, pellets, filaments, and granules. Our study focused largely on microplastic fibers. So these were coming from a fleece coat and are 5 millimeters or smaller in size. And our question was, how do secondary microplastics affect Daphnia magna on the cellular level? So we were investigating apoptosis, which is cell programmed death, and cell proliferation, which is the turnover of creating new cells. We chose to do Daphnia Magna because my project advisor, Dr. Christensen, has worked with this organism in the past. They're ideal to grow under multiple stressors or to put under multiple stressors. They're non-selective filter feeders, and they're easy to grow and maintain in cultures. And this image on the right here is just a diagram showing their simple anatomy, and we were looking at the gut of the animal. They are also, they've been studied in the past with the spheres that were primarily made as spheres, and those are more smooth and rounded, but they found with exposure to that type of microplastic, there is an increase in mortality and decrease in growth and fecundity, or their ability to produce healthy young. And we were wondering, well, what is that connection? Like, what is causing that to happen if it's not chemical leaching or anything like that? So we were looking largely at the physical impact when they ingest it, what damage is being done to their intestine. And this project started around a year ago today with the inception of the idea, and then moving into, after the literature review over the summer, how did we want to go about doing this? How did we want the microplastics to be ingested? How are we going to get that to start happening? So because we were mostly remote in the fall semester, we got a home setup going, and that's the image you see on the right here. And I was able to borrow the stereo microscope and a couple of cultures of Daphnia to begin the processes of getting the microplastics ingested. And they were all non-toxic materials, so it was easy to transfer it to a home setting, which was really nice. And then when we finalized our methods, it's what you see here. So preparing the microplastics, I used a red fleece sweater that was easy to, we wanted the pieces to be easy to identify in the gut. So I manually cut them with razor blades in 1 centimeter squared sections, and just went at it for like 20 minutes with wet media. And then we did a 2-week growing period for the cultures and separated 12 individuals total into half. So 6 individuals were being exposed to plastic and 6 were not. They went under 24-hour fasting period before their 48-hour exposure. And then following exposure, I did dissections of the intestine, and we performed a tunnel assay, which measured apoptosis, and BRDU labeling, which measured cell proliferation. And you'll see in the results that tunnel is going to be red fluorescence, and BRDU labeling is going to be green. So a quick overview here with the results is there was visible ingestion of microplastic in the gut. So on the right-hand side panel here, the figure 1 shows A and B as the control group, and C and D is the exposed group. And the red pieces here are all microplastics being ingested. And it's a lot easier to see once the intestine was dissected away from the rest of the body. The lengths of ingested plastic were congruent with the sizes of algae that Daphnia are able to take in. Apoptosis and cell proliferation were occurring in microplastic exposed groups, and apoptosis ended up being significantly higher in the exposure group. So I'm just going to move into figure 2 here. Part A shows the difference between the control group and the microplastic group in the whole animal size. So the sizes of them were very similar in both groups. And then part B is showing the microplastic fiber length. And the red part is the pieces of plastic that were measured inside the guts. And then the column microplastics are the meat. We took a 1 milliliter sample of the microplastics in the media of the group that was exposed to microplastics, and then evaporated it and resuspended it in combo, which is like artificial pond water. And then we put it onto a slide and looked at it under the microscope and measured it out. and these are a lot higher in length than what was being ingested. Figure 3 here shows A through H of the control group and the exposure group that we measured for apoptosis and cell proliferation. And then I blew up the B, F, C, and G so you could see it a little bit better. It is still a bit tiny for cell proliferation, but the basic takeaway was that cell proliferation was not occurring at all in the control group, but we did see it happening in the exposed group. And if you look very closely at the arrow in F, there is a little green dot, and we indicated that as a positive based on the size and intensity of the signal. This is fluorescence microscopy, which I should have mentioned before, but the fluorescence of green is showing up in this area, and it's the size of the nuclei that would be undergoing cell proliferation. And then for tunnel, the apoptosis occurring in the control group, there was one individual, which is shown here, that did have a positive signal. And then you see it happening more frequently in the exposed group. And especially right here, the signal is very intense. Figure 4 is just showing the data of what we found from each group. So cell proliferation was not statistically significant when done with the student's t-test, and apoptosis ended up being statistically significant. So apoptosis is significantly higher in individuals exposed to microplastics than the individuals that were not. This picture on the left here is some sample pieces of plastic that I took pictures of under the microscope using the bright field view. And you can see that these fibers are elongated, almost twisted, very like blunt, sharp edges in some places. And the shape and size of them resemble that of planktonic algae, which is what Daphnia regularly consume as a food source. And so we made that connection that they are able to take it in, their anatomy is able to take it in easily. And then the plastic retention that is occurring in the gut, previous studies have shown that exposure to plastic has caused retention of that plastic in the gut. So it's staying in there for longer periods of time, even after being removed from an environment where they're being exposed to plastic. And this may also be occurring in our study as well. And this would lead to an increased opportunity for cellular damage to occur that would induce apoptosis, whether it's cellular stress and inflammation response or direct injury. And the increase in apoptosis might explain the increase in mortality and lower growth and fecundity that we're seeing in previous studies. The present study is the first to establish the cellular basis for negative effects resulting from ingesting microplastic fibers in Daphnia magna specifically. So apoptosis and cell proliferation isn't something that was looked at before, and our study was the first to examine that and create this possible link as to why that Daphnia are dying. And then this issue of microplastics and what their impact is on our environment is an infrequently asked question. I was introduced to this topic last year, and it's a newer topic for activists to be discussing, and I think it should be discussed more. And this study in general is just a small puzzle piece added to solving that impossible problem. And where do we go from here? Well, the plastics are made, they're not going away. We need new solutions to mitigating its negative effects. And that might even involve legislation. Future research may be aimed at exploring the intake of other secondary microplastics by Daphnia magna further, whether that's pieces of plastic bottles along with these fibers, like what is their reaction to experiencing multiple microplastic types at once, or it may also be expanding the current study to include food web interactions with predators to Daphnia Magna. So seeing what happens when they're exposed to plastic and populations are declining, what happens to the predators who are losing their food source. I wanted to acknowledge Dr. Arne Christensen for being my project advisor, Dr. Tim Parshall and Dr. Frank Giuliano for being my second reader and honors reader for the Senior Honors Project Committee that I have for this project, and Professor Deanna, Professor Wayne, Professor Caldwell for their support in the honors program, and Gretchen Conrad at Westfield State from the beginning of this project to its completion. And these are my references here for the entire study. It's all previous studies from past exposure studies for Daphnia specifically, and there are also some exposures for other organisms as well. And I'm gonna stop sharing so I can see everyone again, but thank you for listening.
Thank you so much, Gabrielle. Great presentation. So we'll open the floor up for questions. If anyone has questions for Gabrielle.
I just have a random curiosity question.
Sure.
So how was it working on research at home, like on your own? Because I know like for my project, I wasn't able to do it from home for most of it.
Yeah, it was weird. It was so unusual and unorthodox for me because most of my education did take place in the lab in at the university. My professor gave me the resources, and it was more exploratory than anything because our research methods did have to be like finalized in a laboratory setting. So I was lucky to be able to get tested every week to go on campus and do the formal part of the study this spring. And so doing it in the fall remotely, it was definitely interesting. And it felt good to be working on it a little bit at a time and to be able to see like, is this project feasible to do in the lab eventually? And you know, it was more encouraging than anything. So, yeah.
I have another question.
Sure.
So you did it like on a small scale with like the bugs. Do you think like that could translate to like a larger scale with humans, possibly?
That's something I've definitely considered. And I was even discussing this with someone the other day. I feel like microplastics might not have the same effects on a larger scale for humans. So I'm wondering if it would be like a long-term impact versus like this is happening to Daphnia in a very short period of time. So I think it would be something that would have to be more long-term. Like if we're like drinking microplastics, what is that having an effect on long-term?
Yeah, and I'll just echo a question that was asked for the last presentation, but so what do you tell your friends? You know, what do you tell your family members? I mean, do you think we should be changing our behaviors?
Yeah, thank you for that question. I do. It's hard because plastic pollution is so widespread. It's literally all over the world. And so I think a lot of it goes back to the individual to, you know, reduce, reuse, recycle. You know, that's something that I've grown up participating in. And I think it's something that the younger generation has been exposed to more frequently than maybe in past years. But I think it goes to advocating for, you know, reducing our use of plastics and then kind of advocating for the management of waste in a better way to, you know, mitigate its spread into oceans, into these ecosystems that are being impacted the most.
You know, I just want to say, first of all, you know, I'm very impressed that you were able to get some of this research done at home. That's great that you were able to have that, you know, a little bit of normalcy in an otherwise very abnormal situation. I just had a quick question about the Daphnia. Did you notice any like short-term changes in their health? I noticed you mentioned like that they were all about the same size, but were there any noticeable differences between your control group and the ones that consumed the microplastics, even in the short term?
Yeah, so it was more qualitative in looking at it. It wasn't something that we could quantify necessarily, but we did notice that like they were producing neonates at the time. So while they were being exposed to these two scenarios, they were still developing young in their bodies, and because they're all females and from the same cell line, but they reproduce with these neonates, and we noticed that they were like kind of not malformed, but definitely like smaller and like weaker looking, I guess you could say. But compared to the control group, they weren't as like well-rounded or full looking as they could have been. So I did notice that as just like a qualitative thing.
Yeah, that would definitely be another option if you could continue this study,
Yeah.
is to look at some of the health consequences.
Yeah,
Later on,
exactly.
yeah.
And previous studies have shown that with the primary spheres, that exposure to that type of microplastic did cause health implications. So,
Great. Do we have any other questions for Gabrielle? Great. Thank you so much.
thank you.
Thank you so much. So last but certainly not least, we have Hinal Rana from the University of Hartford. And the title of her presentation is The Relationship Between Daily Water Intake and Circulating Apelin, Seeking Feasible Solutions to Curb Population Health Burdens. So I will turn it over to you.
Thank you for that introduction. So I'm going to go on and share my screen. Let's see. Okay, so as mentioned, my name is Hinal Rana. I'm a health science pre-professional major at the University of Hartford, and I hope to soon attend medical school. So I took on this project as honors work, and I'm excited to be able to present it to you guys. So the project is exploring the relationships between daily water intake and circulating apelin as a potential feasible solution to curb population ill health. Okay, so the background. So just a little bit about water. So we know that water is absolutely essential for life, and accumulating evidence has shown that water intake has been related to chronic health outcomes, such as cardiometabolic diseases and acute and chronic kidney disease. So therefore, it's beneficial to analyze the role of water intake within the body. As of right now, we currently don't know how much water individuals should be intaking on a daily basis, but with like the prevalence of disease that we see worldwide and how it affects individuals and their loved ones, the cost-effective health recommendation for water has an extreme value. So one complication when trying to determine health outcomes of hydration behaviors and determining optimal practices pertains to the challenges of measuring hydration status. So as we know, body water balance is complex and dynamic, and so there's no gold standard measurement technique that exists. But there are a variety of biomarkers and dietary collection methods that we use to help us get a sense of hydration status and physiology. So as seen above, there's plasma osmolality, which describes the concentration of the blood. Plasma copeptin, it's the surrogate of the antidiuretic hormone, which basically indicates when the body senses the need to conserve body water. And then we have urine osmolality and urine volume, which also informs the body's need to conserve water, but it also is frequently used to describe fluid intake behaviors. So while there are a variety of ways to measure fluid intake, the most detailed approach that we also thought was beneficial was to measure or have individuals record everything they eat and drink and have specialized software determine the total water intake from all of their foods and beverages. And just before I move on, it's also important to note that total water intake alone is very misleading because it's only one part of evaluating body water balance and its needs. So we're still looking for valuable markers of hydration status and practices, and we're still looking for mechanisms that can link hydration to disease. And so what my project focused on was the novel hormone apelin. Apelin was recently discovered and it's been implicated in chronic health such as like heart failure, hypertension, atherosclerosis, and many more. So apelin was recently found to increase in research in the blood after a single bout and regular exercise in aging animals and humans. So one article that I read showed that the work mechanistically showed that apelin protected and reverse sarcopenia, and so that was a huge finding. However, the work didn't address the potential role of hydration behaviors. And since apelin has been known to play a role in body water balance, drinking more water is thought to increase apelin in the blood and vice versa. It's important to do further studies. So studies have shown correlation between apelin and its receptors in various biological processes. And what we know is apelin's role in body water balance, but we don't know much more past that. So the research is fairly limited. So by expanding on these relationships, we can help researchers get a step further to identifying how the hormone apelin can be upregulated and further improve our health. And so our research lab recently demonstrated that hydration might impair an exercise-induced increase in plasma apelin. So the data was collected at an endurance cycling event just before pre, immediately after post, and 1 hour following the water consumption for like partial rehydration, so post 1 hour. And the figures on the left demonstrated that the cyclists were significantly dehydrated after the ride, which was indicated by plasma copeptin in the middle and urine-specific gravity. And that the water bolus partially rehydrated them, which was indicated by plasma osmolality and plasma copeptin. So this led us to suspect that dehydration might interfere with the ability of exercise to raise plasma apelin concentrations, because we expected to see an increase in plasma apelin at post, as we see right here in the middle. However, it decreased. And we found that water consumption also didn't increase plasma apelin as we thought it would. And so moving forward, we wanted to see the relationship between habitual hydration practices and plasma apelin. So my project specifically, the purpose of it was to investigate and explore the relationships between plasma apelin and hydration markers after accounting for the influence of exercise in those going about their daily lives. So how we did this was we collected data from 78 healthy individual volunteers on our campus, and the population represented various ethnicities. So while half of them were Caucasian, the other half identified as either Black or African American, Hispanic or Latino, Asian, or a combination. And initially, all of the subjects had to complete a familiarization visit where we basically collected all of their demographic information. And they were also educated on how to accurately record everything they ate and drank in dietary logs. So just a sheet of paper where they had to write every single food they ate and every single drink they had. And they were supplied with instructions for the remainder of the participation, which basically was the following week. So the bulk of the participation was over the course of a 5-day observation period where we didn't manipulate anything. And the subjects were told to do what they normally do in regards to their dietary consumption and physical activity, except they weren't allowed to consume any alcohol during their participation. But anything other than that, they wouldn't be judged for. And so as you see in this diagram below, this is kind of like an outline of the entire experiment. So the silverware represented the silverware image represented the recording of their dietary intake. The orange jug represented the collection of 24 hours of urine output, and the blood vials represented the days that we collected the blood samples in the morning. So that was before they ate or drank anything for that day. And yeah, so the urine had to be collected for 24 hours a day for 5 days in a row. So we did supply them with a bag to conceal the urine jug because they had to take it with them to class, to the library, to the gym, everywhere they went throughout the day. And the blood vials were also centrifuged after we collected it, and the plasma was stored in negative 80 degrees Celsius until we measured it. So the Apelin was quantified using an ELISA, which is basically an enzyme-linked immunoassay. And afterwards, after we collected all of our information, the data was checked for normality. I'm going to show the linear regressions that we were performed, and we evaluated the relationships between plasma apelin and the hydration biomarkers, total water intake, and physical activity. So basically everything related to apelin. And then to go a little bit further, we also did tertiles, which I'm going to show at the end, just to analyze the data further based on low, moderate, and high categories using an ANOVA. And I know it was mentioned before in another presentation as well. So the results. So we started to analyze the linear regressions, and we found that plasma apelin was not related to total water intake as seen on the left, or even to exercise, which is on the right, and that basically opposed any previous literature. So we originally intended to perform complex analysis to determine the role of total water intake by first accounting for the role of physical activity, but there was really no reason to conduct that analysis because plasma apelin was not related to water intake nor physical activity. And so we were surprised to find that apelin was not related to exercise. And this honestly could have been due to reporting errors. So yes, we gave ample instructions and reviewed exercise reports daily. But since this was self-reported exercise, some of the individuals that participated might have underreported their actual exercise practices or even overreported if they forgot to chart. And so similar reporting issues could have also been caused by the lack of relationship to total water intake, or apelin might just be a function of the body water balance, so inputs and losses rather than what someone is actually taking in. And so plasma osmolality was also not related to plasma apelin along with copeptin. So the lack of relationship with plasma osmolality could have suggested that the concentration of the blood was not a stimulus for apelin release. And alternatively, the plasma osmolality is known to correct itself fairly quickly with short-term changes of water. So it might not even be a perfect biomarker to measure regular hydration behaviors from day to day. Plasma copeptin, on the other hand, is able to detect regular hydration behaviors and seems to be a better body measurement. So apelin was not related to copeptin either, which was kind of surprising. And then for the last two, plasma apelin was also not related to urine hydration biomarkers such as urine osmolality, which is the concentration of the urine, or urine volume. So in theory, urinary markers are used to tell us the amount of, it's used to tell us more about body water balance. So if we're well hydrated, we produce more voluminous and less concentrated urine. And that demonstrates less of a need to retain water. But our data collectively suggested that circulating apelin is not a sensitive biomarker of hydration status or practices. And so based on these findings, we decided to do tertile groupings just to view the data in a different way before we made any strong conclusions. And so as we see in these graphs, the tertile groupings show low, moderate, and high groupings of each of the independent variables. So while there's no significant differences amongst the means, both total water intake up in the left-hand quarter and plasma osmolality demonstrated a potential dose response, which basically means that it appeared to be a stepwise increase in the tertile groupings, with increasing plasma apelin. So that potential dose response could have suggested that our sample size of 78 individuals was not sufficient to observe the relationship between apelin and hydration. So in other words, our investigation could have been statistically underpowered with the amount of individuals that we were studying. And so in conclusion, circulating apelin did not seem to be a sensitive biomarker of hydration status or total water intake. And while we were probably statistically underpowered, if it takes a large group of individuals to detect a difference, if one exists, then that still doesn't imply a great utility for hydration assessment in individuals. So, but to confirm this, again, with the potential recording errors that we might have faced, future work should intervene and manipulate like hydration statuses to confirm that there is a lack of relationship between plasma apelin and hydration. And I think that this work should also be continued because, as I mentioned earlier in the presentation, since we're facing rising and substantial health issues, quality of life, and economic burdens, the cost-effective practice such as consuming water can really impact individuals on their day-to-day life. And so I just want to conclude this, and I want to thank my research advisor, Dr. Colleen Munoz, and two more individuals, Dr. Claudia Oakes and Dr. Donald Jones, who have really pushed me every single year of my college career to do the best I can and to successfully be an 18 credit honors student. And I'm going to stop sharing my screen so that I can answer any questions.
Thank you so much. Thank you so much. So we'll open the floor now to any questions.
I have questions.
Go ahead.
First of all, I think we can all just join in the amazement of how little we know about hydration, which seems so simple. But is there a takeaway in this Hinal about, I mean, can any conclusions be drawn about how people can know if they're drinking enough? Any recommendations about how much we should be drinking? Or was this just too many degrees away from that?
Yeah, so I've actually looked this up before, and I've researched an optimal amount of water to intake, and it's shown that on average, women should approximately consume 2 to 5 liters of water a day, and men should aim for 3 liters per day. But obviously, if you sweat a lot throughout the day, you should consume a little bit more than that. But another indicator for hydration is by looking at your urine. So you should aim for like a lightish yellow color urine, but if it looks like water, that's too dangerous. So your urine should always have a tint to it.
Thank you.
I have a couple of questions. One is just a practical thing. How did you get all of these students to volunteer for this study, particularly, you know, at an undergraduate level where you're not only having them collect urine, but you're taking blood samples from them? Is this something you're doing with a graduate program, or is this strictly undergrad?
It was strictly undergrad
Wow.
students, so age range like 18 to 21 or even 22. And I think one of the leading factors for their participation was they were given money for participating, especially because they had to wake up every morning and draw blood, and they had to carry on a jar of urine throughout their entire day. So, you know, it was the little things that I think money plays a big factor in allowing individuals to go about their daily lives and help with research. So I think that was just a motivating factor to having individuals carry on urine.
Right. And just another, again, a practical question. Who did the blood draws for you? Did you have people that were trained?
Yeah, there were students that were phlebotomy certified and even like professors. So they were the ones, it wasn't only me that did the research, it was a team effort. So there was a huge group of individuals that did different aspects of the project, especially when inputting all the dietary logs. I know that I did that over the summer. and I was able to take the computer home, but it took so long to get through one individual's 5-day period and input every single food they ate, and try to determine how much hydration was coming out of that food.
Great. And just one more quick question. I know this is a novel hormone, but do we know where it's produced? Where does it come from?
So as far as I know, apelin is within our body, and it's a water balance hormone, and it's shown up in the blood. I think that I would have to do further research to see where it is actually produced. So I apologize that I can't
No.
answer.
That's okay. That's okay. I appreciate, your honesty. That's not knowing is a perfectly valid answer. So good for you.
And there's very limited research on apelin,
Of course.
and that's why we're trying to study it. And again, I think that we would have to continue to do research on it to see, even with a different population, to see if apelin is really not related to hydration, because there could have been some reporting errors that might have shown no relation in it. Some biomarkers might be significant to apelin that we might not know of. So I think we just need to keep doing research on it to find out more.
Great. Any other questions from the floor? Since we do have a few minutes left, I'm just going to throw out a general question to all the presenters. Number one, is there something that motivated you to do this particular research that you chose? And does this relate to your long-term plans? Or is it just kind of a stepping stone? I would be interested to hear. So maybe we'll start with Anna.
Well, this kind of just came about. Dr. Connors is, well, I considered her to be like a good professor to me. Like we catch up after class and stuff. She's on sabbatical right now, but like I'll still text her all the time. But I really just did it to do it. And I mean, my plans are to go to medical school. So it doesn't really relate with bacteria and Roundup, but it was fun to do, so.
Gabrielle?
For me, what sparked my interest was actually reading an article about microplastics, and because I was gonna go like microbiology route. That is an interest of mine and something I kind of want to explore after I graduate. But when I read that article, I realized how like big of a problem it was and how little I knew about it. So I really wanted to delve into that, and I'm glad that I did pursue it. I like started having conversations with the biology department, and it led me to Dr. Christensen and his work with the water fleas. So it just like all came together so perfectly. But now I am considering maybe doing something in the ecology. So I'm gonna explore those options.
Great, thank you. Hinal?
So I've been doing research with Dr. Colleen Munoz on dehydration for a while. So that kind of sparked my interest in focusing on hydration for my research. But I also want to go to medical school, and I want to be a physician, but I want to focus on the heart. So knowing that the hormone apelin has like effects on the cardiovascular system, kind of like helped me combine hydration and apelin in order to further investigate how this hormone can alter or prolong health effects.
Excellent. Does anyone have any other questions for our presenters or comments or anything they'd like to add to the discussion?
Just congratulations to everyone for, this is all extraordinary research, but I just have so much respect for how hard it was for all of you this year. Nothing was easy, and I know that you all overcame so many obstacles and barriers to get to where you are right now. So congratulations to you all.
Yeah, I'll second that. And I think Vanessa, Diana also agreed. I think, you guys are resilient. This has been a tough year on multiple levels, but here you are, presenting, your research at an honors conference, and something you really should be proud of, an accomplishment, and something no matter what you do in the future, whether it be medical school or go into ecological research, or maybe you do something totally different, but this is something that you can always look back on as a positive experience in your life at a time when things didn't always look so positive around us. So congratulations to all of you. And, I hope you enjoy the rest of the conference. Just a couple of pitches for the rest of the day. So we do have another session coming up shortly at 2:30. So hopefully you'll be able to join us for that session. And faculty and staff, we do have a happy hour later. So please join the NRHC board for a nice little happy hour to kind of relax and de-stress a little bit. And students, there is a dance party tonight at 8 p.m. on Zoom. So get your 80s gear and be ready to party it up with our student rep, Taylor Bennett, who is just wonderful. So I guess that's about all I have to say. Did we get, I just want to make sure we had any questions in the chat room? No, I think we're good. So if we have no further questions, then I will wish you all a wonderful day, and thank you all for being supportive of NRHC, and especially of this conference that has certainly been something new for all of us. So thank you all for your attendance and your support, and have a wonderful rest of the conference.
Thank you.
Bye, everybody.
Congratulations, presenters.
Yes, great job, everybody.
Yes.